Revise with Dr. B. This is week 2, organisation part 1. So this week I'm going to go through the first half of organisation, the section, and I'm going to look at digestion and digestive enzymes. How this works. So I'm going to go through the main points of the topic, not everything, just some of the important bits. You'll be given signposts to further resources, particularly videos on the required practicals, past paper exam questions, and I'm going to be doing weekly videos all the way through to the exams, so please do subscribe. Another added thing I'm going to add this week is some exam practice, so I'll tell you a bit about that when I get to it. So firstly, there are 5 levels of organisation. So you have cells, tissues, organs and organ systems that make up an organism. You will need to know what a cell is, what a tissue is, so it's a group of similar cells that have similar structures and functions, and organs, groups of tissues working together to perform a specific function, organ systems, groups of organs working together, and then obviously organism is all those organ systems working together. So here is an example. Okay, so if we look at the heart muscle cell, and then you've got heart muscle, which is made up of tissue, and then heart is made up of several types of tissue to make an organ, and then that's part of the circulatory system that makes the organ system, including the lungs and the blood vessels. The digestive system, you'll need to be able to label the digestive system, so if you want to pause and maybe make some notes at this point, that would be useful. So first required practical you need to know is the required practical on food tests. So it's using reagents or chemicals to test for carbohydrates, proteins and lipids. And if you go to the video that's in the box below, then that will give you more information. And also I've got some exam questions on that coming up. So digestive enzymes. Well, the first thing is you need to know about the lock and key theory. So the lock and key theory of enzymes states that the enzyme has an active site and a substrate has a complementary or similar shape, not the same shape, but it's similar and it's complementary, and it fits into the enzyme active site, it collides with it to produce an enzyme substrate complex, and then the enzyme splits those substrate into products. Here's an exam question, and it's all about the lock and key theory. So it says use information from the diagram above in your answer. It's 3 marks. So the kind of things it's looking for are here. So substrate does bind to the enzyme as they are complementary. I probably actually would mention that they're complementary to the active site. So then the substrate is broken down into products, and then the products are released, and the enzyme itself is not changed and therefore can be reused. You'll need to know where each of the enzymes in the digestive system are made. So for example, carbohydrates, so amylase for example, produced in the salivary glands, pancreas and small intestine. It breaks down starch into sugars or glucose, and this takes place in the mouth and small intestine. Protease, so a couple of examples are things like pepsin and trypsin. You might see them in exam questions. It's produced in the stomach, pancreas and small intestine, and they break down proteins into amino acids. Lipase, so lipase is produced in the pancreas and small intestine, and it breaks down lipids or fats and oils into fatty acids and glycerol, and it takes place in the small intestine. So what they're doing, the enzymes are converting food into small soluble molecules, and they are then absorbed into the blood in the small intestine. Okay, what can these products that are produced by enzymes be used? Well, it can be used to build new carbohydrates, lipids and proteins. Some glucose could be used for respiration. What's the role of bile? Well, a couple of things. It's made in the liver, stored in the gallbladder, and it's squirted. I don't like that word, squirted. It's kind of secreted into the small intestine, and it's after the food has been in the stomach. And it has 2 functions, and you will need to know these functions. So bile neutralizes acid from the stomach because it's very acidic in the stomach, and it needs to be alkaline, and it's needed for the enzymes that are released into the small intestine to work. It also emulsifies the fat or lipid droplets from large to small droplets. So it's really key that it's not breaking down anything. It's just making those droplets smaller. So that increases the surface area for lipase enzymes to break fat down. You'll also need to know about the effects of pH and the effects of temperature on enzymes. So if we look at the effect of temperature first, you can see that at low temperatures enzymes work quite slowly, so the rate of reaction is quite low, and as it increases, the rate of reaction increases because you're getting more kinetic energy that's moving the molecules around faster, and therefore the colliding, so the substrate and enzyme collide with each other more frequently. You then get an optimum temperature that can be different from different enzymes, but this is where the reaction is working as fast as possible. The enzyme is then at high temperature is denatured, and we'll talk about denatured soon. It's a very important word, but basically the enzyme stops working. pH, slightly different. So the enzymes in the digestive system, they'll only work well at certain pHs. Okay, so they have different optimum pH values. So usually proteases that are found in the stomach, their optimum pH is around 2, whereas in amylase, for example, in salivary gland, in the saliva in your mouth, it's neutral at pH 7. So denatured. So if we look at the top diagram, you can see that usually the substrate will fit, and they have complementary shapes. But as temperature is increased far above the optimum, what's happening is that the enzyme's active site is permanently changing shape. So therefore this prevents the binding of a substrate, so stopping a reaction. The next required practical is investigating the effect of pH on enzyme activity. So they're usually testing how amylase digests starch at different pHs. So that's using the reagent iodine, and I've done a you'll be able to have a look at my video. So again, the video is in the text box below. Okay, so you're going to need to know the required practicals. I'm going to give you for free now some exam packs, and also I'm going to put the links in the box below the video. So what follows is a series of exam questions. So I'm going to show you all the exam questions, and I'd like you can pause please, and then all the answers are at the end of the video. So here goes. Okay, guys, I hope you found that useful. Next week, I'm going to be doing the second part of organisation. So keep on revising, keep at it, and you'll get there. All right, see you soon. Bye.